Start early? Why not? We'll start early. I only came up to pour water for Kay, fortunately, since we didn't have a glass at the time. So, welcome to the Royal Society and to the annual Croonian lecture. As many of you know, this is the this is the premier lecture in biological sciences of the Society. It's named for William Croon, one of the founder fellows of the Society, and it was first given in 1738. I'm John Scahill. I'm biological secretary of the Society, and it's my pleasure this evening to introduce this year's Croonian Medal winner, Kay Davies. Kay is Dr. Lee's professor in anatomy and director of the Medical Research Council Functional Genomics Unit in Oxford University. Her research involves the molecular analysis and development of treatments for human genetic diseases. As you know, one of the most topical areas in medical research. And her lecture this evening is on the genetic neuromuscular disease Duchenne Muscular Dystrophy, and it's called From Diagnosis to Therapy in Duchenne Muscular Dystrophy. Kay.
Thank you very much indeed. Can you hear me? Yeah, good. First of all, I'm delighted to be here, and thank you very much to the Royal Society for giving me this opportunity. And I'll say it at the end, but I receive this award on behalf of all the people that have worked for me over the years. And what I'm going to do is take you for a journey from the genetic diagnosis of DMD, which 30 years ago didn't exist, through to what is now the most exciting phase in DMD therapy. In fact, DMD really has led the way for approaches in many other diseases as well, and I'll mention some of those as I go along. So I'm going to emphasize the importance of patient registries. When we began this work, we were much more concerned with just the diagnosis of individual patients. If you're going to do any sort of translational research, you really need to have close organizations like the Duchenne Muscular Dystrophy charities that can actually know very well the natural course of this disease in order to know whether your therapy is working. I'll talk about precision treatment and what that means as individual patients, as well as what it means to deliver a therapy worldwide. Different modalities, I'll go through each of the ones that are working at the moment and in clinical trials, indeed, for DMD, and finally talk about combination therapy. So, for those of you not familiar with this disease, it's truly a devastating muscle wasting disease. It's a common disease, relatively speaking. It's early confinement to a wheelchair about 12, and many of these boys do not actually present a diagnosis until 4 or 5 years of age, unless there's been a previous family history. The patient population in the developed world is estimated to be about 50,000. And what's interesting is when we began this research, no biopharma was interested in looking at rare diseases, and now they're tripping over themselves to try and get into this space. And still today, there is no curative treatment available, and lots of sort of supportive approaches. And in DMD, there's one approved therapy, and I'll talk about that in a bit more detail later. It's a particularly interesting disease because it's one of those where even if you could screen every pregnancy, you would not eradicate the disease because there's a very high new mutation rate. And perhaps what is also interesting is that sequencing the human genome still does not tell us why that is the case. So you can ask me that question, but I won't be able to illuminate you. So this is the tragedy of this disease. These are 3 brothers in the same family, and they are born before the first diagnosis. This is, you can see the boy aged 2 years on the left there, looking reasonably healthy. And this is his brother at the age 10 and his other brother age 12, and the severe muscle wasting that you can observe. So when we started in 1980, there's unreliable carrier detection. No prenatal diagnosis was available. But were there any clues? Well, the first clue that you may have picked up from my slide is this disease affects males only. And if it affects males only, that means that it's on the X chromosome, because the normal X chromosome in females can compensate for the dysfunctional gene on the abnormal chromosome. And these female patients, many of them characterized by John Edwards, had a break in this region of the X chromosome, so we had a clue then that DMD was localized here. But there was also an anomaly because these were mild form of the disease which showed great similarity, first characterized by Becker, which was thought to be linked to color blindness, which happened to be at this end of the X chromosome. And at this time, this work was pioneered by Bob Williamson. In fact, I was working in Bob Williamson's lab, and it was Peter Harper who had collected all the families that allowed us to ask the question, can we find DNA markers for this part of the X chromosome, and can we establish once and for all that this really is where the gene is localized? And so we set about with Brian Young in Glasgow to purify human chromosomes. Now, of course, you don't need to do that. You can just choose your bit of sequence out of the human genome and go straight for it. But we were able to isolate a whole series of different DNA markers here that surrounded this putative DMD locus. And sure enough, in collaboration with our international collaborators, I'm not going to go through them all, but this was indeed an international collaboration, we were able to confirm that DMD was indeed at XP21 on the X chromosome. What about Becker muscular dystrophy? Well, Peter Harper's group then went on to use the same series of markers to establish that, in fact, BMD is not linked down here, but in fact is an allelic disease. So you have one disease with two very different phenotypes. DMD, the boys go into a wheelchair at 12, are normally dead in their late 20s, some of them earlier than that, and it's clinical management that has pushed the survival rate of these patients from their teens into their 20s. BMD, by contrast, the cutoff point is diagnosis at 16, but I'll show you patients that were not diagnosed until their 40s, and some of them live into their 80s. So the gene, having been established at XP21, was finally localized and cloned by Ron Wharton, who identified the breakpoint of one of those female patients and was able to identify the gene. And Lou Kunkel, based on a deletion of a family, was able to identify sequences which led to the identification of the gene. And it turns out that this is quite a remarkable gene. It encodes a very large messenger RNA, but this isn't the largest messenger RNA in the human genome. If you think of hemophilia A, the gene for factor VIII is 9 kb, so it's smaller. The gene, however, is remarkable in the sense it's 2.4 million bases of DNA, and if you compare that to an E. coli bacterial genome, which is 4.8 megabases, this is the largest gene known in the human genome. That, in part, of course, will explain the high new mutation rate, but it doesn't entirely, and I'll come back to that. The corresponding protein was called dystrophin by Lou Kunkel because the lack of it causes muscular dystrophy. But the real remarkable thing is 65% of these patients have deletions, and they can be just a couple of kilobases or they can be several megabases across that region of 2 megabases. And so Tony Monaco proposed that the difference between DMD, that very severe disease, and the mild phenotype was whether that deletion took out part of the gene that allowed you to have that gene back in frame. So you ended up with a smaller, partially functional molecule, the so-called open reading frame hypothesis, and that's turned out to be true and indeed exploited in therapy. So what about the organization of this gene? And this indeed also turned out to be remarkable. It's made up of lots of little pieces, 79 different segments, which are called exons, for those of you not familiar with molecular biology. And these are spread over 2.4 megabases of DNA. And they're not uniformly spread. There are very large introns that are several hundred kb, and some of them that are just a few base pairs. And as it says here, this encodes a 424 kilodalton dystrophin protein. And that's just indicated schematically down here. I'll talk about the structure of it in a minute. There are also isoforms of this. Oops, sorry. Sorry, I better get it back. There are other isoforms of this gene, promoters within the main 14 kb transcript that give rise to smaller transcripts of this dystrophin gene, but so far no functional relevance clinically has been assigned to these, although they may be associated with the cognitive deficits in this particular disease. Now, 30% of patients are known to have some form of mental impairment, but I'm not going to discuss that in any great detail during this talk. What we do know is the mutations don't seem to be related to where you have deletions in that gene. What is also very interesting is that in spite of not knowing what the DNA sequence is that drives this high frequency of deletion, there clearly is a preferential deletion region, and this is demonstrated here. These are the 79 exons of the gene. This is the work that Sue Forrest did when she was in my lab, and you can see that there's a peak around exons 6, 8, 10, and a large amount about exon 3 here, but most deletions are in this region between exons 44 and 55. Oh, and I should just mention that this deletion profile exists in all populations studied. This is the paper that I put at the bottom here, is the latest in a Chinese study indicating this is a disease that affects every population in the world in exactly the same way. So what does dystrophin do? For those of you not used to looking at muscle, this, whoa. I'll get used to this in a minute. Pointer, right. Antibodies raised against the protein identified dystrophin as a protein at the muscle membrane. This is it. You see the uniform labeling across all these uniformly sized muscle fibers here. This is the milder form of the disease, Becker muscular dystrophy, where you can see that there's only partial labeling, and this is very typical of a Becker patient. Partially functional molecules don't bind the membrane as well as the full-length dystrophin. And this is the picture in most DMD patients. There's complete absence of the gene. Now, occasionally, you will see a revertant fiber where naturally there's a skip of the mutation or the deletion. If it's small enough, it puts the protein back in frame, but it's not enough to rescue the DMD phenotype. So DMDs localize at the muscle membrane, and in fact it's binding to a very important complex at that membrane. So it doesn't lie across the membrane itself, it binds to the dystrophin-glycoprotein complex. It's first characterized by Kevin Campbell and Ozawa's group, and it binds the actin, F-actin in the internal part of the cell and laminin in the extracellular matrix. So it's a very important link between that extracellular matrix and the F-actin in the internal part of the cell. If you take that dystrophin away, you actually remove that very important link. You then lose the components of that complex. They're no longer stable, and you get the muscular dystrophy that we observe. And this is just showing you a schematic, a very important link. And what this also led to is that if this hypothesis coming from Kevin Campbell and Ozawa's group was right, then mutations in these components of the disease should lead to muscular dystrophy. And since the genes are on the autosomes, it should be autosomal linked muscular dystrophy. And indeed, that is exactly the case. And some of the therapies I'm going to talk about may well be applicable to the so-called limb girdle muscular dystrophies. So dystrophin really is a shock absorber, if you like. And this is just a schematic that was drawn out by John Tinsley, where if you assume that that's the muscle membrane, if you bounce a lot on that on your trampoline, then you will come back to the original alignment. If dystrophin is missing, then you don't. So when the gene was cloned in 1986, we were all very excited. Just like when any other gene is cloned, everybody said, well, therapy will be around the corner. So just note, that was 1986, and even if the field is accelerating, it still takes a very long time. But it was a real challenge. This is a large protein. Not only that, it's a large structural protein. So it's not like cystic fibrosis, where you can replace just a small percentage of the CFTR mutated protein. You have to replace at least something of the order of 40% of normal levels to get clinical benefit. Now, there may be slightly less than that, and of course you may get some efficacy, but if you really want to cure, you've got to get up to those sort of levels. Not only that, you need to target all muscles, skeletal muscle, the diaphragm, the heart. Eventually you want to target the brain to deal with the cognitive deficits. If you look in the literature, you can see also that dystrophin is expressed in smooth muscle. We don't know what dystrophin does in smooth muscle, so we really need something that will target all muscles in your body. We need, muscle takes up more than 40% of your body mass, so we need systemic delivery. So we've got systemic delivery that's got to be safe. We need to avoid the immune response. So our heart sank in some ways because this is something that will need a lifelong treatment. We've got to avoid the immune response. It's a structural protein, and we've got to get a lot of it back into the cell. So I'm going to cover the genetic approaches only today. That's not to say that drugs that modulate the immune response, fibrosis and so on are not relevant in this disease. I'm just not going to cover them because those sort of therapies will be used in conjunction with the genetic therapies as we go through the next 5 or 10 years. What I am going to cover is the therapies that target the DNA, the RNA, and the protein level. And the first of these is called exon-skipping. And as I said to you before, you get a mild phenotype if you can get those exons stitched together to leave the protein in frame. And this is just one schematic showing that in a patient. If some segments are missing, they're not joined properly, they're out of frame, as you can see here, and therefore no dystrophin is produced. You can persuade the cellular machinery to exclude yet another segment, which then leaves you with, if you take out exon 46, in this case, you can join 44 to 47 in frame, and you get a functional, partially functional protein. Now, what function you get back will depend on what structure of that rejoined dystrophin is, and I'll come back to that in the context of gene therapy at the end. So how do we induce exon skipping? This was first pioneered by George Dixon's lab and the Japanese Matsuo's lab. And what you do is you get an oligonucleotide that can mask the splice machinery that is adjacent to your exon 46 in this case, that will then persuade the molecular splicing machinery to skip past that exon and give you what you require, and that is a partially functional but shortened molecule. Now, I don't have time to go into exon skipping in great detail, but I just want to highlight that there is an approved drug, and I'll come to that in a minute, known as eteplirsen, that is produced by Sarepta Pharmaceuticals, which skips exon 51, which is applicable to 13% of patients. However, the approval of that particular drug was controversial because in some patients it works well and in others it doesn't, and it's not particularly efficient. And so a lot of companies now are looking for other DNA chemistries and multi-exon skipping to try and improve on this. And this is just illustrated here. This is work we did in collaboration with Aurélie Goran-Valley using tricyclo-DNA, and this is compared to the 2-methyl and the morpholino DNA chemistry, and this arrow here just indicates the exon skipped exon. This is in the MDX, the mouse model of the disease, which has a stop codon in exon 23. And you can see the improved exon skipping efficiency here with the tricyclo-DNA compared to the other chemistries. If you look in the brain in this particular situation, tricyclo-DNA also exon skips, crosses the blood-brain barrier, and you can see that very clearly in the various sections of the brain here. Now, as far as I know, none of these others actually do go across the blood-brain barrier, but a lot of these have now improved, even past what I showed you in that slide. Etoplirsen, as I say, for exon skipping is now approved. Other exons are in development for exon 44, 45, and 53. However, every time you target a different exon, you have to set up a new clinical trial, which is another multi-million-pound enterprise. So that is challenging in itself, which means the cost of the drug at the end is going to go up. You need to target the heart, and as far as I know, I'm not sure that etoplirsen does target the heart very efficiently, but there are new antisense chemistries coming along, not just changing the DNA backbone. Matthew Wood's group, for example, are putting peptides on these morpholinos to target it more efficiently to the heart, although they're not in clinical trials yet. Wave Life Sciences is changing the stereochemistry of the DNA, which greatly improves the efficiency, and tricyclo-DNA I've already talked about. CRISPR DNA editing, and Jennifer Doudna gave this lecture last year, so I assume you all came to that, so you'll know everything about editing. But I'm sure most of you have heard how revolutionary gene editing can be, and what we could do is think about just editing across all those exons that are commonly deleted in patients, which is between exons 45 and 55. And that would target 63% of patients. So one trial would demonstrate a clinical advantage for all of these patients. The problem is we still have to get CRISPR, non-target effects, etc., sorted out before we can go down that route. But there are still several companies that are multi-exon skipping using conventional approaches. But just to remind you, of course, this will only convert DMD to BMD. And just how you put those exons back together and what structure you end up with in the dystrophin molecule is still not a precise science. So another promising approach, which only applies to 13% of patients this time, is full stop codon read-through. And this has been pioneered by PT Therapeutics. And the idea here is that you use a chemical which will persuade the cellular machinery to read through that stop codon and give you a full length molecule. And the product that is in the clinic is known as Translarna or Atalurin. Oop. Shouldn't have done that. Oh. And unfortunately, Translarna did not meet the endpoint of the phase 3 trial, but there are a lot of patients now that have been treated with this drug, and if you look at the overall statistical analysis that's been published, there does appear to be a subsection of subgroup of patients that do respond to this drug, but they don't respond dramatically. But nevertheless, these boys can walk faster. So this drug, because of its lack of clinical efficacy, is not approved by the FDA, and it's conditionally approved by the EMA in Europe. So I think it is clear that it does work for some patients, and it doesn't for others, but we don't really understand why. So what I've just shown you is personalized medicine in the sense that you have to know exactly what that target mutation is before you can decide what is right for your particular DMD patient. But there are two approaches which apply to all patients, whatever their mutation, which makes them particularly powerful. And the first one I want to discuss is gene therapy. Now, gene therapy has come a long way in the last 20 years. In fact, there's almost one new biotech company a day that's formed to do gene therapy, not of DMD, but of some disease or other. And they are worth billions of pounds because the big pharma are now moving into gene therapy because it's such a targeted and efficient way of delivering genes back into patients when they have genes that are missing. The problem for DMD, of course, is that we had a 14 kb message, and something like AAV, which is the virus that is most commonly used for delivering things to muscle, is only got a capacity of just under 5 kilobases. So that was a challenge. But during our analysis of these numerous Becker muscular dystrophy patients, the late Sarah Bundy sent us this particular gentleman's DNA and said that he presented in his 40s. This is him just after he joined the army. This is him just after he'd walked up that cliff, but he needed the walking stick. And for those of you very familiar with the disease, you can see his characteristic gait like that in this photograph here. He was mobile at his 65th birthday party, and he died in his early 70s. What was absolutely remarkable was that he has 46% of his coding region completely missing. So how could that be? Well, the first is it could possibly be because he's got some sort of genetic compensation, because he's been fortunate to upregulate a compensatory gene or a series of genes. The other is that, in fact, 46% of dystrophin is enough to give you a partially functional protein that allows you to be as mobile as this gentleman. And so we went back to Sarah Bundy, and it turned out that he was a member of the family here. That's the proband that I've just shown you the picture of, patient 324. And he had a distant relative who agreed to have a muscle biopsy here, who was a bodybuilder, age 28 at the time we did this analysis. So the question we had to ask was this patient producing a truncated protein? And this is the antibody study, and you can see at the N-terminal region, which binds F-actin, and the carboxy-terminal end, which binds that complex of proteins across the membrane that's so important, are intact, but he's missing for this bit. So that was highly suggestive that he was indeed producing a partially functional truncated protein. And in collaboration with George Dixon, we did, and I think actually Nick Wells may have been part of this study, we did the transgenic, we reconstructed his gene, we introduced it into the MDX mouse, and we were able to prevent the pathology in the MDX mouse. So the truncated gene then opened the way to use this mini-gene to treat muscular dystrophy. Fast forward another 15 years. This is the original patient that we described, and these are a whole series of different, now called micro-dystrophin genes, because even the mini-gene, if you want to put a promoter in front of it to make sure it's muscle-specific, then you still don't have enough room. So you have to nibble away at this mini-gene to get microgenes and test these in the MDX mouse in the hope that they will be able to give you sufficient functional benefit. And this has really been pioneered by Jeff Chamberlain and Dewan's group and Zhao Zhao's group in the States. And the various genes are summarized here, but you can see some of them have the hinge regions, which are marked here. This is an important binding site, which binds nNOS, which is thought to be very important for DMD patients. Having said that, there are a lot of Becker muscular dystrophy patients that are deleted for that region that don't have that nNOS binding site. So whether that is really important is somewhat controversial. Which hinge region is important, we simply don't know. And Jeff Chamberlain has recently published a paper where he's tried out a lot of these different mini-genes, and it's clear that no one of them is perfect. So it's a compromise. So there are now 3 clinical trials ongoing. National Children's Hospital, this is the construct they use, doesn't contain the nNOS binding site. This is Solid Biosciences, which does. And this is Pfizer's construct here. So I'm sure they will all work to a certain extent. But what we can also say is Sarepta have published data, not in the literature, but on their website, which shows a DMD patient running once it's had several months after the administration of this virus with no adverse events. So this is potentially completely transformative for the therapy of muscular dystrophy. The problem is, will we have an immune response? And in fact, the first gene therapy that was carried out in DMD was by Jerry Mandel, who's did the work with Sarepta, and he did a local administration of the AAV. And what he observed was that some of the patients had a pre-immunity to AAV, so some of them, about 20% of patients, will not be eligible, and redosing may not be possible. So, in summary, this is an incredibly exciting result. Pfizer are doing 2 doses, and the analysis is expected in 2019. They say that their data at the moment look very similar to those of Sarepta, but they're not actually talking about their data. Sarepta are now moving into a phase 3 trial, placebo-controlled trial, because of course these patients, well not of course, but these patients are on steroids, so you have to bear in mind that some of the positive effects that you see will be due to that steroid administration. But look at this, 81% increase in dystrophin. That has never been seen with any exon-skipping protocol. It's usually of the order of 10 to 20%. But I think the secret here is when you do an AAV delivery, you deliver to all muscle very efficiently, not just to one muscle type. And I think the exon-skipping protocols that we've been looking at in the past have really preferentially gone for one muscle or another and don't target the heart very well. Having said that, the mini-genes, the microgenes that I've just described to you, don't work in the heart in the MDX mouse very well either. So it may be that this is the first generation of microgenes, and when we see the data, we may have to go back and generate different microgenes that may be able to target the heart. Anyway, coming back to this rather spectacular increase in dystrophin, these patients showed a dramatic decrease in their creatine kinase levels in the serum, which is indicative of muscle damage. That had not been seen in any muscular dystrophy trial before. And they targeted patients in a very fine age group, 4 to 7 years of age. Solid Biosciences did a dose escalation study. This is a huge dose of 10 to the 14 viral, is the viral titer here. Solid Biosciences decided to do a dose escalation study, and they included patients 6 to 17-year-old. And their initial data, which was released some weeks ago now, didn't show very significant levels of dystrophin. And so we're now hopeful that when Solid Biosciences go on and do the next higher dose, that they may be able to see a difference. And you can see that in dogs. There's a dog model of Duchenne muscular dystrophy. If you do 10 to the 13 viral titer, you don't get much effect. If you get 10 to the 14, you get a dramatic effect, and you get these dogs running down the corridor. So there's clearly a threshold above which you have to get that viral titer in order to get a result. But what this means, of course, is if it only works between 4 to 7 years of age, it is not going to be generally applicable to all patients. The other problem is if you go in too early, then the muscle will grow very rapidly and you'll get loss of the virus and you'll have to try and re-administer. If you go too late, there may not be enough muscle to transduce, so you won't necessarily get rescue. But I think we have to remember, we don't have to cure these patients. We have to improve their quality of life. And so it may still be possible that if we can get the titer high enough in this older age group, that we might be able to transduce in the muscles that are very important for their arms, for example. If you ask a DMD patient what is very important for them in their lifetime, they will tell you it's the point at which they lose their independence in a wheelchair. If we could do something about that without curing the whole disease, it would make a big difference. So there are challenges, as I've already said. There is an immune response. There will be problems with repeat administration, and people are now trying to develop nanoparticle delivery for the microgenes instead, and perhaps if you've got a nanoparticle, you may be able to put the whole dystrophin gene back. The other problem is the preparation of high titer virus is a challenge in itself, and there is not enough viral production in the world to cope with DMD, let alone haemophilia and all the other diseases. Sustainability of effect, as I've said, you may gradually lose, And it's interesting, in the Independent today, the cost of a AAV therapy for spinal muscular atrophy is $2 million. So we have to come out with another paradigm of how we're going to pay for these types of treatment in order to be able to make sure that we can get it to all the patients, even if we can get enough high titer of virus. So, is there another way in which we might be able to work this therapy for Duchenne muscular dystrophy? So we've been always very interested in whether there is indeed a compensatory protein. This was really started when we first identified that patient, and we thought, well, maybe he might have a compensating gene. The other inspiration was David Weatherall, who was always telling me that fetal hemoglobin one day would be able to compensate for beta globin in adult thalassemia. Fetal hemoglobin is what it is. It's a fetal gene. It's turned off, and then the adult gene is put on. It could be that there might be a fetal gene for dystrophin. And so while we were trying to get the full-length cDNA for dystrophin, Don Love in my lab identified a protein, well, a DNA sequence, which didn't map to the human X chromosome. And that's shown schematically here. This is what we call utrophin, and we called it utrophin because it's ubiquitously expressed. And this is the full-length dystrophin that I've shown you before. These two proteins are more than 96% homologous in what I call the business ends of the molecule, that binds F-actin, and that binds the complex at the membrane. And it's this central rod domain where there's much less conservation. But don't forget, in that patient with had most of his, 46% of his gene deleted, he was deleted across this region here. So there may not be as much evolutionary constraints for this region here, which would explain the differences. For those sharp people, you can also see that utrophin is slightly shorter than dystrophin, but it still covers a megabase of genomic sequence. It's an ancient genomic duplication event. So the question is, if this is so similar, are there any clues that this might be able to compensate for dystrophin? So the hypothesis was that it's so simple, going back to that schematic here, we could use dystrophin or utrophin here and be able to treat the disease. Well, there are some clues, very similar to the fetal hemoglobin story. If you look at human development, dystrophin and utrophin are present at the sarcolemma, the muscle membrane, together in a 12-week fetus. As you go towards birth, dystrophin expression increases until you get what I've shown you previously. But as you can see here, utrophin is progressively downregulated in muscle. And in fact, it's only localized at the neuromuscular junction by the time you're born. So their regulation is interrelated. If you look at normal muscle, there's dystrophin, and there's DMD muscle, and that's an exon-skip fiber there. But there's utrophin, and these are neuromuscular junctions here showing that utrophin is uniquely in adult muscle at the neuromuscular junction. In the absence of dystrophin, you get some spreading of utrophin from the neuromuscular junction onto the muscle membrane. We now know that this is not a transcriptional upregulation, but a stabilization of the protein because it's able to bind that complex of proteins. But it's certainly not enough to compensate for what's seen in patients. I'll forget about that. So we have a continuous cycle of muscle damage. We've got a mature, healthy fiber with dystrophin on it in a normal individual. And in a DMD fiber, you don't have any dystrophin, and then you get the progressive muscle degeneration. If we can find a way of modulating utrophin expression to take it back to the fetal form, then we have an opportunity to compensate and make this much more like a mature, healthy fiber than it otherwise would be. So how much utrophin do we need to compensate for dystrophin? And these are studies carried out in the MDX mouse by John Tinsley predominantly, and Rosie Fisher, and Sarah Squire, and I look across all the people sitting in front of my group there, Ben Edwards, and Ali Potter, utrophin transgene prevents dystrophy in the MDX mouse. We can cure a mouse by increasing the levels of utrophin. And you probably can't see this from the back of the room, so you'll just have to believe that's a much healthier fiber. It's much more obvious when we inject it into the DMD dog over here, where you can see the not healthy muscle here, and then the much healthier muscle on this side. So less than twofold provides some benefit in the MDX mouse. And of course, if you're going to administer a small drug, you then have to ask the question, if you take a pill, you're going to upregulate utrophin everywhere. Well, actually, if you upregulate dystrophin everywhere in the mouse, you don't get an adverse effect. And sure enough, we demonstrated, again using transgenics, that if you overexpress in the mouse, we didn't see any deleterious effects. And that obviously is very important. The other observation was if we took full-length transgenics, so-called Fiona and Freddy, the response was very dependent on having that uniform expression in all muscle types rather than a little bit in one particular muscle. And that's very similar to the paradigm that you see with AAV versus exon skipping. And this is the percentage of centrally nucleated fibers in the EDL muscle and the diaphragm. And you can see in both cases whether you have a full length or Freddy at much lower levels. You can see with both full length at different levels you get rescue, slightly less rescue in the diaphragm in Freddy. And then if you look at normalized force, which is another way of mentioning it, again you can see even with the low levels of expression, you're still getting movement towards the normal phenotype. So this was the time when we really had to think about setting up a biotechnology company, because you can't do any of these clinical trials without doing that. And I'd just like to acknowledge this was a partnership between Muscular Dystrophy UK, Summit, and the chemists at Oxford. And I was a chemist once. The other thing that has happened in the chemistry space over the last decade or two is that you use very clever chemistry now. You don't have to screen 5 million compounds. You can use intelligent chemistry, and therefore there is a hope that you might be able to find the needle in the haystack, but I'm not going to go into the intricacies of that. So we set up a phenotypic screen where we could identify those sequences in the promoter region of the utrophin gene, and that's shown here, just with two promoters, the so-called A promoter and the B promoter. This is only expressed in endothelial cells, so we were particularly interested in those expressed in muscle. And Kelly Perkins in my lab has identified several other promoters in this region recently, which would have been contained in this construct. So what we did was we took the gene, I put it together with the luciferase gene, and then screened those compounds, those chemical compounds from the chemists Angie Russell and Steve Davies' group, to try and identify anything that increased the levels of utrophin. And the sequence that's very important in this is this N box. Chanju's group a long time ago identified the acetylcholine receptor transcriptional regulation and identified this particular N box configuration that determines the localization of that expression at the neuromuscular junction. And that is exactly what utrophin has and dystrophin does not. And that explains why utrophin is found at the neuromuscular junction, where it becomes, this becomes the predominant regulator of that gene. And in early human development, this must be overridden because you get the expression of the sarcolemma. So azutramid was the drug that we identified, which increases utrophin levels in cell lines. We asked the question, well, if we put this in vivo in the MDX mouse, this is the control, and this is a normalized force. This is exercised MDX, and this is exercised MDX plus azutramid, where you can see we are indeed having a dramatic effect. So exercise measured by the force grip, protected against the loss of function otherwise seen with exercise. And we could demonstrate, I'm not going to go through it all today, that this was correctly localized exactly where dystrophin is localized at the muscle membrane. We then went on to do a clinical trial, or rather Summit went on to do a clinical trial last year And what we did was we administered the drug and then did muscle biopsies at 24 weeks and 48 weeks. And the blue staining here is developmental myosin, which is a measure of muscle regeneration. So you can see that after 24 weeks, this was the baseline, and this was at week 24, that appeared to be it was a very dramatic difference. Unfortunately, at 48 weeks, that particular reaction was not sustained, and Summit had to discontinue the trial. The drug was shown to be safe. There were no adverse effects, but it clearly was not going to show us the efficacy long-term that we needed to see. So we decided, particularly in view of the work that Kelly had done on new promoters, that instead of just taking a small section of the gene, we'd actually try and use CRISPR to introduce the luciferase into the gene in frames. So this time we had the whole genomic context for those of you that are aficionados of transcriptional regulation. And so we identified a new screen which would introduce both utrophin and utrophin B. It was actually in exon 7 here, the so-called Lucy MDX line. And we used that to screen molecules. Did it identify molecules that we wouldn't otherwise identify? And the answer to that is yes. So this is the original H2K line versus the LuMDX. And so what we're now looking for when we do a screen is for compounds that are uniquely expressed in this particular cell line, but not that, because this is the line from which azutamid came. If we have a compound that upregulates utrophin here, we would hope the mechanism of action is going to be slightly different. We're now at a stage where we've got a lead candidate. So azutamid was stopped because of the non-efficacy, as I've already shown. The so-called next generation were related to azutamid and showed efficacy just as azutamid did, but we couldn't find anything that was non-toxic, and also we wanted something that had a different mechanism. So we're now, actually this has moved into further down the discovery line, we now have a lead which we hope we'll be able to take to clinical trials in the future. So coming back then, where are we? That should be 2019. I didn't notice that. Where we've got gene therapy, which gives you a truncated protein. We've got a truncated protein from exon skipping, but we've got a lot of improvement of efficacy needed to be able to make that a series of FDA-approved drugs. Stop codon read-through. There's just the atalurin from PT Therapeutics. Whether other drugs will come along, there are certainly other companies that are working on that particular strategy. CRISPR, of course, will be a single correction early on. And early work from Charlie Gersbeck, actually he reported it just recently, suggested that the CRISPR AAV delivery actually gets into the satellite muscle cells. Now that's important because it'll be repopulation if you start to lose the virus. So that's extremely promising, but of course CRISPR is still some way from the clinic. But nevertheless, this is I think a real step forward if we can target the satellite cells, and modulation of utrophin, as I've already said, is there. So the point I'm trying to make from this slide, though, is each one of those is just short of a cure, maybe give you 50% compensation. Can we do combination therapy? So since we only had the utrophin tools and we collaborated with Matthew Wood's group with exon skipping, we asked the question, if you put utrophin back into muscle, would it displace dystrophin? Could we do it in an additive fashion? This is just a paper that's in Press that Simon Gouillet did with Ben Edwards and Sarah Squire and Aaron Babs and Lee Moore in the group. This is MDX, utrophin, dystrophin. That's the merged. That's when we add the peptide PMO, morpholino chemistry. And I can't read it from that slide. And what you can see, you probably can't see it from the back of the room, but you can see that they do co-localize at the membrane. But what is perhaps more important is this part of the slide. This is the force drop for MDX. This is the PPMO, which partially rescues, and this is the full-length transgene MDX, which also rescues, but not back to wild type. If you administer these together, you get a wild type level. of functional benefit. So that suggests, in fact, therefore, that you might be able to put utrophin back in if we can get the drug into the clinic. We might be able to use that even for Becker muscular dystrophy patients because it doesn't actually interfere with the binding of dystrophin. And what we also demonstrated was that actually if you overexpress, we're not going to get these levels normally, but if you overexpress utrophin in the wild type, we can make it function even better. So there are hints that actually dystrophin and utrophin don't do exactly the same thing in exactly the same way, and we know that from their sequence, but that we can certainly, if we combine utrophin therapy with any of these other therapies, we ought to be able to get closer to that cure. So in summary then, genetic approaches plus combination therapy, I think will eventually get there. And it's an incredibly exciting time in DMD research. So I'm just going to finish with, of course, we've now got potentially transformative therapies. So do we therefore need to do newborn screening? And I think the answer has to be yes, because the earlier you get in with any of these therapies, whether it's SMA, DMD, whatever, you will always get a better effect if you catch the disease early. So I think we'll have to now think about the relative efficiency of being able to newborn screening. And indeed for SMA, that has spinal muscular atrophy, that has already begun because AAV delivery of the SMN gene in spinal muscular atrophy also gives dramatic effects. But these SMA kids are born floppy, and unless you get in very early, you're not going to get sufficient efficacy. So I leave you with that health challenge. So in summary, we do need patient registries of all of the diseases in order to be able to monitor very carefully how these patients are doing. DMD is very heterogeneous. It's been quite difficult to get very good endpoints for some of these clinical trials, which is why we have to have placebo-controlled trials at the end. Precision treatment versus treatment of all patients. Obviously, we want to treat all of them. And I think the FDA are beginning to move now to getting shorter clinical trials where it's the same type of treatment, even though the exon target is slightly different. Clearly, the way forward, particularly in a complex environment like a disease like DMD, you need to try exon-skipping, read-through gene therapy, as well as small molecules. And finally, I think this will be the paradigm for all of these diseases. We need combination therapy. So I finished there. I can't put all the name of the people that have worked in my lab, but the ones in bold are some of the people that are sitting in the front who do all the hard work and keep me sane. These are the people in Summit that we wouldn't have been able to deliver the clinical trial without, and we learned a lot from that clinical trial, so we're well set up to do the follow-up, and the chemists who do all of that medicinal chemistry in order to be able to us to get those leads that will eventually go into the clinic. Last but not least, you can't do this research without the cooperation of the patients, the families, and the charities. And just a big thank you to all of them. And actually also to the MRC. There are many times when people said gene therapy for muscular dystrophy will never work. And the charities and the MRC have always believed that eventually someone would get there, even if it wasn't us. And I think we are now in a position in the next 10 years where we'll see these transformative therapies making a real difference to our patients' lives. Thank you very much.
Other questions? Yes.
Well, thank you, Kate, very much. It's somewhat humbling to hear that if I was sharp enough to ask a $1 billion question, I'd only get 6 months of treatment. But you tantalizingly said that one of the features of Duchenne's is this very high rate of new mutations. And this is not accounted for by the size of the protein. Do you have any ideas or thoughts about what is the driving force for that?
It must be something to do with the chromatin folding. If you take STS deficiency, which is another X-linked disease, there are inverted repeats. That's another high level of deletion. Actually, it's true of haemophilia also. And you can identify those inverted repeats. There's nothing in the DMD locus, but it's such a large gene, and the way the chromatin loops out must have something to do with the way this gene deletion is promoted. Yes. And interestingly, it doesn't happen in the mouse. At least there are several mutations in the mouse, but not as many as you might think if it was at high mutation rate.
Get it. There's a thing. Thank you.
Very much. Oh, thank you. I was going to ask you anyway about the satellite cells, and you just mentioned it once, I think, at the end. I wonder how many of the treatments were active on the satellite cells, and is that something that if you worked on and treated early, that might just be a great help?
In many of those cases, they haven't looked at the satellite cells. In the other AAV studies that have been published, they don't transduce the satellite cells. The CRISPR technique that Charlie Gersbach has used does target the satellite cells. It works much more efficiently in neonate mice than it does in adult mice. But that paper's only just come out. I think people are just beginning to realize that, well, they knew that that was going to be the case, but you have to get proof of principle first. And I think now we have to go back and decide what constructs are there. Are there new viruses that we can, new constructs of the virus even, that might promote that rather than the present generation? Most of the companies are using AAV9, but not all of them. And there may be differences between those viruses on how they transduce the satellite cells. And I think that will also feed out in the clinical outcome that we'll get from these trials at the end of this year or the beginning of next.
Please repeat.
Thank you, Kay. That was a marvellous lecture. What would a screening program look like in practical terms?
I think they'd have to incorporate into the sort of Guthrie spot. There'd be things that they'd put in that you'd have a DNA test that would cover most of the common disorders like spinal muscular atrophy, cystic fibrosis, because that's another one where there now is an effective treatment for certain patients, and DMD. I mean, it has been tried in DMD. There was a screen. I think Peter Harper's group in Cardiff undertook one, and so did the group in Holland. But the patients were ambivalent, the parents were ambivalent as whether they really wanted to know in advance whether their child was going to suffer from muscular dystrophy when there was no effective treatment. So it wasn't carried forward. Now we're at a point where we've got potentially effective treatment, particularly for boys born today. I think it'll shift. So one single DNA test it's the cost that will prevent it really, but now there's an opportunity, perhaps that will come into clinical practice. There's a really big push in Belgium in particular for the spinal muscular atrophy one.
Thank you.
The second row back there. Oh, sorry.
Okay, thank you for that, all that genetics. You mentioned immunology and immune responses as potentially blocking therapy. Do you have any comments to make on that as to what extent it's been looked at and what's been found?
Well, there's both a T cell and a humoral response. The problem is the capsid of the AAV, and again, that depends which virus you used. So they're now just beginning to look at that. And the people that manufacture these AAV viruses are also looking at it. In fact, there must be hundreds of different AAV capsids that you can now use at there's whole gene therapy companies that are producing different cocktails of AAV. So that'll be very important. Jerry Mandel did report antibodies against the dystrophin protein, but whether that is clinically significant, I think, is a debatable point, and you may be able to do immunosuppression for that. So I think people will be more careful and will start to do the sort of experiments people like you as an immunologist would want them to do, now that we've got the tool and the material to do it with.
Yes,
okay, fantastic. Thank you. So a comment, we recently got permission from the Neonatal Screening Committee to take a punch from the official Guthrie card blood spot to test for genetic risk of type 1 diabetes to randomise into an oral tolerance trial in newborn children. So there is light at the end of that tunnel, and we were surprised and delighted, and that trial is up and running. But my question is, given the length of sustained effort and funding and perseverance, 1986 or whatever onwards, what single thing would you do differently? Was there a garden path or a blind alley?
Thank you, John. It would have been useful, I guess, to collaborate more when it got to the therapeutic area, because I think we could have. There was collaboration, and there is, PTC Therapeutics helped us set up the trial we set up with Summit. But I think if we'd have had a little bit more crosstalk on that, we might have got combination therapy, we might have learnt a bit more, and going to Liz's point, we might have learnt more about the immuno. But every one of these things has depended on a technological breakthrough of some form. And so, although it's 1986, it's doing this. So the more we can exchange ideas, and I think the spinal muscular atrophy, the fact that they went intrathecally with the SMA, the SMN gene, in very young babies and got a dramatic effect, enabled the FDA to think these viral delivery is safe, relatively speaking. We should try it in DMD. And I think it's, so I'm not sure I would have done anything differently, but I think the field is really, and the regulatory authorities are moving fast now because they've realized the importance and the unmet clinical need. They're keeping it safe, of course, but they're learning from other diseases. So it's not just progress in DMD that will get us to the finish point. It's everywhere. And I think that gives us patients even more hope.
Yeah. You got one? Yeah, I got you. Red pullover? Yeah, good.
Thanks, Kay, and good to see you. I have two questions, basically. Going backwards, earlier and earlier, would you contemplate going into the fetus? Because there you would solve delivery problems, you would solve amounts problems, you would have less immunity problems, and perhaps sustainability. And coming to sustainability, is AAV the right vector? It doesn't integrate, yes. It hangs around, and how long do you think will it work, and will that be a problem that you have to readdress?
Right, a complex question. I think it's a question of balance. I mean, if you could get a sufficient efficacy when the child was born, obviously you wouldn't do fetal gene therapy. I think this is probably the wrong disease to do fetal gene therapy, because I think there are probably signs of the disease, even quite late stages of pregnancy. We've never looked at it, but I suspect that will be the case. There are other viruses and non-viral routes that are now being developed, and that will be the way forward. AAV is there because of the transduction efficiency in muscle, and we needed to get proof of principle, so that's where we are. I think you'll see coming down the pipeline, as I said, there are lots of biotech companies that are totally dedicated to producing new viruses, and maybe one of those will be the safe virus that we hope we're going to be able to get.
Yes.
Sterile inflammation. Is this sterile inflammation, and can you interfere with the destruction?
That's not being looked at in any great detail, so the answer is I don't know.
Well, the reason I'm asking is if you do the MDX model, you've never seen so many macrophages recruited in a hurry.
Yeah, yeah. People, I mean, the problem is people haven't really looked at that because the MDX, I haven't mentioned it, but I mean, if I bought an MDX in here, you don't scream, but it would be running up and down because it's so mildly effective, relatively speaking. So I don't think we've had a good animal model to look at that, but they could do that in the dog model.
That's it. I mean, complex, but at a wonderful stage of development. So good luck with the next phase.
Thank you.
Good. Thank you very much.
Thank you.
Well done. It was very good.